In the name of Allah, the most beneficent and the most merciful. Assalamu alaikum wa rahmatullahi wa barakatuhu. How are you, dear students? I hope you all are fine and well. I welcome you in Pakistan International School Types virtual learning system for the session 2020, and we are studying biology for grade first year. Dear students, today's chapter bioenergetics. As you know that we are going to continue this topic and this chapter, and we will continue the previous topic photosynthesis, and in that we will study today dark reaction. So let's get started. Dark reaction, or that is also named as light independent reaction. So in light reactions convert solar energy into chemical energy, like they produce ATP and NADPH. NADPH called as reducing energy and ATP called as assimilating energy. Assimilating power and reducing power. And these two powers are utilized in Calvin cycle or in dark reaction for the synthesis of glucose. So Calvin cycle makes sugar from carbon dioxide. We know that till here, like we have finished light reaction, there is no use of carbon dioxide. Now carbon dioxide is utilized in the Calvin cycle. So what happened in this reaction? ATP generated by light energy, light reactions, they provide energy for sugar synthesis. NADPH produced by the light reaction provides electrons for the reduction of carbon dioxide to glucose. And in this cycle, which are the steps and how this carbon dioxide change into glucose, we will see in dark reaction. Actually, this is the reaction in which light is not required, but it doesn't mean that it is done in the dark reaction. It can be done in the dark reaction if the plants have sufficient amount of assimilating and reducing powers. But as they are forming sugar in the Calvin cycle, so they are utilizing ATP and NADPH altogether. So this reaction is wholly solely depends on light reaction, but it doesn't need light directly. So what happened in this reaction, we'll get just summary, that this reaction creates a 6 carbon molecule, which then splits into 3 carbon molecules. This part of cycle is called carbon fixation, and as we know that here carbon dioxide is going to be utilized in the reaction. So that is actually called the carbon fixation process. So let's talk about a Calvin cycle, that is also named as a C3 cycle, or that is also named as PGA cycle or G3P cycle, because as well as Calvin cycle or dark reaction or light independent reaction, there are lots of names of this. So in C3 cycle, the first stable component which is formed by this reaction, the first stable compound which is formed here, that is having 3 carbon in that. That's why this is called, that is 3-PGA. So that is called C3 cycle. Okay. So it is composed of 3 stages, stage 1, stage 2, and stage 3. What are these stages? We will just talk about in a few minutes. So Calvin cycle is termed as the series of metabolic reactions in which carbon dioxide is reduced to G3P. Here is G3P, and these reactions explored by Melvin Kelvin and his co-worker in the University of California. So for this purpose, Melvin Kelvin got the Nobel Prize in 1961 for this work. Kelvin cycle can be divided into three phases: carbon fixation, reduction, and generation of carbon dioxide acceptor. And carbon dioxide acceptor is ribulose bisphosphate, which will be formed here. So this is here ribulose 1,5 bisphosphate. It means it is having two phosphate groups in that, so that accept carbon dioxide, and that carbon dioxide gets into the Kelvin cycle, and then by utilizing of ATP and NADPH here and ATP here, then we get sugar, as well as again we are getting carbon dioxide acceptor. So let's talk about different stages of carbon fixation. Cycle, Kelvin cycle. So first we'll talk about this first stage, in which carbon dioxide is accepted by ribulose bisphosphate in the presence of ribulose rubiscose, that is the enzyme which is which performs its function here by the conversion of ribulose 1,5 bisphosphate into 3-PGA. So let's talk about here. So first we will see here three molecules of carbon dioxide and 3 molecules of ribulose bisphosphate RUBP in the presence of rubisco ribulose bisphosphate carboxylase or oxygenase. Its name is given because this reaction is going on in like there's we can say the temperature sensitive. It is very temperature sensitive. So in some plants that acts as oxygenase, in some plants it acts as carboxylase. Carboxylase means that it just add carbon dioxide with ribulose bisphosphate, and oxygenase means it adds oxygen in place of carbon dioxide. So this happens in C4 plants. Like here, the first stable compound, we are having here 3-PG, but in C4 plants there is 4 carbon compound. This is 3 carbon compound. You can see 1, 2, 3 carbon atoms are there, but in C4 plants we are having 4 carbons in that compound. That is the first stable compound in C4 plants. So if that functions as oxygenase, so that is called a C4 cycle, not C3 cycle, and that can also be said to be Het-Slack cycle. So these plants which perform this like C4 cycle, they are called C4 plants. And for example, some grains, rice, and like wheat, like which plants are just grown in a very hot climate. So common plants are C3 plants, means when they do Kelvin cycle, so the first stable compound is a C3. It means it is having 3 carbon in it, that is 3-PGA. 3-PGA. Like at carbon number 3, that is 1, 2, 3, at carbon number 3 we are having glycerate is having phosphate group. So let's get started about carbon fixation. That when carbon dioxide enters, there must be any host to just accept that one. That is called carbon dioxide acceptor. So that is ribulose bisphosphate in the presence of ribulose bisphosphate carboxylase oxygenase. That in the presence of this enzyme, that RUBP accepts carbon dioxide. RUBP is having 3 molecules, and then 3 molecules of carbon dioxide are entering inside the that Calvin cycle. So in between these areas, like transferring from the entering of carbon dioxide up to 3 pg, there are some other intermediates which are formed here, but they are unstable, and soon they get changed into 3-phosphoglycerate or 3-phosphoglyceric acid, or that is called 3-PGA or PGA or 3-PG. So this blue triangle, you can see that is carbon fixation process. So here carbon dioxide, which is having 1 carbon, so it will add or react with 1,5-ribulose bisphosphate, that is 5 carbon atom. So actually 1, 2, 3, 4, 5, this is 5 carbon atom, this is 1 carbon atom. So it will become 6 carbon atom, in which there are 6 carbon atoms is present. So short-lived 6C intermediates, but there is no intermediate shown in this diagram. So this 6 carbon compound that is broken into 2 atoms or 2 molecules of 3 carbon. And so 6 carbon molecules split into 2 3C molecule known as 3-PGA, PGA, or 3-PG, phosphoglyceric acid or phosphoglycerate. But in this reaction, you are just, you can see that here we are having, we are getting 3 carbon dioxide, like 3 molecules of carbon dioxide. So we multiply everything with 3. So here 6 carbon compound is broken down into 3 carbon compound. So but there are 3 and 3 6 like 6 molecules of that. So when it is broken into half, so that will be 6 molecules of a 3 carbon compound. Okay, so that reaction type is synthesis, like a synthesis of 3-PGA is going to be done here, as well as enzyme also, which is functioning here, synthetase, like the enzyme which helps in the formation of the product, and that is Robisco, and energy is absorbed here. That is not energy releasing process. Let's talk about phase number 2, that is reduction. So now you can see here this is Kelvin cycle. This one is the Kelvin cycle. So now what happened in this, like we have done here carbon fixation, and from here to here there is a reduction process. Reduction, you can see here, reduction means reduction of the compounds which are going to be formed in the next phases. (3-PG) or 3-phosphoglycerate then receives a phosphate group from ATP, which changes into 6 molecules of 1,3-bisphosphoglycerate. This makes 6 ATP, now turn back to 6 ADP. Here at this position, this is carbon fixation. And now from 3-phosphoglycerate up to 1,3-bisphosphoglycerate, what happens here? There is reduction. So here ATP is utilized. Now this energy which comes from light reaction, now this is utilized here. So because these molecules are 6, so 6 ATPs are utilized to make 6A DP plus, what is that? Yes, 6 inorganic phosphate. So this energy which is received by the breaking of phosphate bond here. So we know that 7.3 kilojoules per mole, kilocalories per mole is that. So that is utilized to change this 3-phosphoglycerate. And then what happened in that, one more phosphate will be added in these groups in this molecule. So on carbon number 1, 2, and 3, now we are having 1,3-bisphosphoglycerate. So 1,3-bisphosphoglycerate is reduced by gaining the electrons from NADPH made in the light-dependent reaction to 6 molecules of glyceraldehyde 3-phosphate G3P. A phosphate group is also released during reaction. Energy from breaking of the bond used to make G3P. Now this is very, very important because here is the compound which we need to make glucose. So 1,3-bisphosphate by using NADPH. This is the reducing power which is which breaks its bonds. So there will be these hydrogen bonds by which this 1,3-bisphosphoglycerate is reduced, and it is changed into glyceraldehyde 3-phosphate. So in this, phosphate group is released out. Actually here this when these 6 phosphate, these are not released here, sorry for that. These are like bound with bisphosphoglycerate to make it 1,3-bisphosphoglycerate. Then by reducing power of NADPH by adding the electrons from NADPH, what happened? It is changed into glyceraldehyde 3-phosphate, like 6 more phosphate groups are here we can say P inorganic phosphates, 6 inorganic phosphates are released here. And a phosphate group is also released during this reaction, and energy from breaking of the bond, which is by breaking of this hydrogen bond, so that is utilized for making G3P. So this process in which this is this compound is reduced into G3P, that is called reduction process. And here one more thing, that G3P, now it is 6 molecules. One molecule will be released from here, that is 3 carbon compounds. So one molecule, you can see here, one molecule is released, that is also G3P, and that is utilized to make glucose. So actually this is the main theme of this reaction. So here what happened, there are okay, so out of these 6 molecules, 1 is released out and 5 will move to the next reactions, because next reaction which we can see, that is RUBP, regeneration, or that is called regeneration process. Ribulose bisphosphate, that is carbon dioxide acceptor. So in this, a series of complex steps that require the carbon skeleton of 5 molecules of G3P. Here we know that here that is 3 carbon compound, but at last we have to get 5 carbon compound. So how it will be formed? converts these G3P molecules into 3 molecules of ribulose bisphosphate. So ribulose bisphosphate, the carbon acceptor of carbon fixation process. So cycle spends 3 more molecules to just convert this G3P into this 5 carbon compound. This is 3 carbon. So here 5 threes are 15. So here 5 carbon and 5 threes are 15. Number of carbon is same. No addition, no releasing, but that is having the different structural formula. So that becomes ribulose 1,5 bisphosphate. Here 3 ATP molecules are utilized, and then it will become 3 ADP, and that phosphates which are removed from here also. One is utilized to just enter water inside it, and that is, sorry, one is added here in this one. Okay. So we can see here 5G3P, which is here, that is changed by using of 3 ATP into 3 ADP and phosphate group. So that is changed into 3 ribulose bisphosphate. For the next net synthesis of 1 G3P sugar, the Calvin cycle consumes 9 ATP. So how it is happened, we will see in the next slide. 6 molecules of NADP and makes 1 glucose of sugar. So now you can see the review of this all reaction altogether. So first of all, we will see about carbon fixation. So that is what? That is incorporation of carbon dioxide or carbon into organic compounds by living organisms, chiefly by photosynthesis in green plants. So that is consumed inside. So this one, this whole is carbon fixation. Okay, next one, if we see reduction. What happened in reduction? 6 molecules of a 3-PGA react with ATP, 6 ATPs, and is transferred to 3-PG. In these 3-PGA molecules are in green plants, they changed into 1,3-bisphosphoglycerates. 1,3-bisphosphoglycerates. Okay. So here 6 ATPs are utilized, and that is changed into 1,3-bisphosphoglycerate, and then later on it will change by utilizing energy of NADPH by breaking down the bond of hydrogen. So hydrogen will add in this 1,3-bisphosphate in place of phosphate. So phosphate will be removed, and this will become G3P. Okay, the next one is regeneration of carbon dioxide acceptor. If we see this darker blue, that is regeneration process, in which this G3P changes into 5 like into ribulose phosphate, then into ribulose bisphosphate. So from here, that will ribulose phosphate, then from ATP, there is like phosphates are added into ribulose bisphosphate, like at carbon number 1 and 5. So that is called 1,5 ribulose bisphosphate, and then that ADP is removed. 3 ADP is removed in that reaction. So here water is also added. Okay, so this is regeneration of carbon dioxide acceptor with the help of Rubisco enzyme. So if we see its overall reaction, so we can see that overall reaction is carbon dioxide, 3 molecules of that, which are utilizing 6 NADPH molecules and 9 adenosine triphosphate molecules, and then we'll get G3P, 3H2O, and from 9 ATPs we are getting 9 ADP and 9 for inorganic phosphate, and from 6 NADPH we are getting 6 NADP. So energy utilization is there, not energy formation. One more important thing, this sugar, this is actually here, it is not sugar, it is G3P1 molecule. But we know that sugar is a 6 carbon compound. So from where the 6 carbon comes? Because this is 3 carbon, 1, 2, 3. So once the carbon, this Calvin cycle moves on, so it will make 1 G3P. When this carbon cycle moves twice, so then it will form a complete glucose molecules in which we are having 6 carbons. And by glucose we know we the plants can make other its carbohydrates like cellulose, like other starch and like that. So it can form these organic compounds from glucose. So it means when this Calvin cycle moves twice, then we can have one sugar molecule. So for twice, and to just moving on twice, we can consume 12 NADPH and 18 ATPs and 6 carbon dioxide molecules in that reaction. So that is so complicated, and which is having lots of enzymes also functioning for this purpose. And so, dear students, with this summary, we have completed our topic, Calvin cycle in photosynthesis. So, inshallah, we will see our next topic, respiration, in the next chapter and the next lecture as well. Wish you all the best. Allah Hafiz.